A very big hello to my cutest little champions out there. How are you all? I hope you're doing absolutely great. Well, here I am with the one short lecture on principles of inheritance and variation, my dear children. And after this, you will be getting your all 1 marker question, and after that you will get the 2 and 5 marker questions. And with that, I can guarantee you that you will get the 13 marks that you could get easily from this particular chapter. Okay, I promise you that. But you have to promise me one thing, that you will be very consistent with the videos that would be lining up. I know there might be 50 minutes video, this and that. But my dear children, if these 3 hours that you're going to spend on me, my dear children, if at all it guarantees you 13 marks, don't you think it's worth it? Yes, definitely it's worth it. And if at all you feel that way, if at all you are ready for this challenge, then you have to comment down in the comment section that I am in for the 30 days challenge. Let me see who all are actually in and who all are gonna take the whole 13 marks with them during examination. So I'll be making this video a bit fast, not as fast as in you may not understand anything, but to extent that it can be covered in a very limited time and you will get all the important information as well. Now, if at all you are someone who wants detailed explanation of things, I have made the whole chapter in Sankalp series. You can watch the parts out there, but I'll definitely recommend you to try this once. It will be definitely helping you really well. So starting off with our lecture by introducing Mr. Gregor Mendel. But why do you need to know about him? That's the question. Well, because he was the one who experimented from 1856 to 1863, that is for whole 7 years, my dear children. And he was experimenting on what? He was experimenting on Pisum sativa, which you also call as garden pea. Well, why did he do the experiments? So that we get to know more about genetics. And you know what happened at the end of these 7 years? Well, these years are very important for you to note down. 1856 to 1863. Yes. So at the end of all these years, he finally became the father of modern genetics. And not just that, he gave us so much information about how genetics work in us, my dear children. And so super fascinating. So you should be here with me to listen that out. But ma'am, at the first place, why did people believe him or his work? First thing is that he was the first person to use math in biology. Suppose I come here and tell that 10% of this is happening and so many things, you will obviously believe me. Whenever you give mathematical evidence and show that, my dear children, people believe you. So that was one thing. Second thing, he used large sample size. It was not like I will do experiment on 2 things and that's all. That's how I'll get the result. No, he had large sample size. Now the third thing, it was not like one time I'll do the experiment or twice I'll do the experiment. He repeated his experiments over generations, my dear children, proving that whatever he was trying to prove is actually correct and it's not a random thing that is happening out there. And then the fourth and the most important thing, he used a plant which had clear and contrasting traits. What do you mean by that? Contrasting traits as in they are opposite. For example, violet and white. These are something opposite and I can clearly see it. Now, if I do the experiment on that, I'll be able to understand, okay, that it can be white or probably violet. So there were 7 contrasting characters which he took and you need to remember all of them. You'll get MCQ questions. Which one is not the one that Gregor studied? So you should know the 7 pair of characters. Remember, there are 7 pair of character. Each one of them you call this as a trait. So 7 pair and you call them 14 traits. 7 pair. 7 has, all the pairs has 2 things. So 7 into 2, 14 traits were being studied. What were those? So you can start with flower. Okay, there is flower color and flower position. Flower color, violet is the one which is dominant or which you call as purple, and white is the one which is what? Recessive. Now, if we talk about flower position, there is axial. That means, for example, from a point where a branch should be coming off, there a flower is coming. This is axial position. Terminal position, for example, at the tip of something I am getting a flower, that is terminal. Which one is dominant? Axial is the one that is dominant. A comes before, so A is dominant. You can remember like that. Okay, so flower color and flower position. Then comes what? Your pod color. For example, if you talk about green peas, it comes inside a pod, my dear children. This pod, which one do you choose? Is green or yellow? you will choose something that is green. So green is the one that is dominant, okay, and yellow is the one that is recessive. Pod shape, if we talk about the shape, will you choose something that has full or which is very dry and constricted? You will obviously choose something that is full, inflated. You call this as inflated. So in pod shape, inflated is the one that is dominant and constricted is the one that is recessive. Now comes to seed color and seed shape. So usually we have seen the green seeds, right? But that is not the one dominant. Yellow is the one which is dominant out there, and green is the one which is recessive. Now if you talk about seed shape, it can be round, that is nice, right? So that is dominant, and the wrinkled one is what? Recessive, my dear children. So flower color, flower position, seed color, seed shape, pod color, pod shape, and now you're left with one thing, that is plant height. One is tall and another one is dwarf. So 7 pair of contrasting characteristics or 14 traits in total. That's what you want to you have to remember. Now going ahead with the next thing is that he did his experiment. And how did he do his experiment? He took a tall plant and he took a dwarf plant. Ma'am, what do you call this condition where both of them are capital T, capital T? This is known as homozygous condition, where both of them are the same thing. Now this is a chromosome, this is a chromosome. From a pair of chromosome, only one chromosome will be going to a gamete, my dear children. Similarly from here, only one will be going. But either way, small t will be coming, here also capital T will be coming. So whenever you join both the gametes, you get capital T, small t. The first generation of plants, my dear children, it will look like this. What do you mean by that? T is dominant over small t, which is why all of them are tall. So they are tall, but their genotype is what? Capital T, small t. This is the thing of F1 generation. Now in F2 generation, Mendel was not able to, not satisfied with what he did. So he thought, we will cross this. So whatever progenies he got over here, he thought of crossing it. And what did he find at the end, my dear children? Let's see that. So over here, he took 2 progenies, and this is known as Punnett square, my dear children. So he took 2 progenies having capital T, small t. So one capital T, small t over here, one capital T, small t over here. This and this cross, you get capital T, okay, that means they are all tall. This and this cross, you get capital T, small t, they are still tall. Why? Because T is dominant over the small one. This and this cross, you get capital T, small t, again you get tall. This and this comes, you get one dwarf plant. So if you are asked about the phenotypic ratio, phenotypic as in the physical appearance. So how do they appear? 3 of them are tall and 1 of them is dwarf. So phenotypic ratio is 3:1. What about the genotypic ratio, ma'am? So 1 is capital T, homozygous tall. 2 of them are heterozygous tall. Hetero means they are 2 different things, right? So 2 of them are heterozygous tall and 1 is dwarf. So what is the genotypic ratio, my dear children? Genotypic ratio is 1:2:1. 1 capital T, 2 capital T, small t, and 1 small t, small t. Over here the phenotypic ratio is mentioned. 3 of them are tall, 1 of them are dwarf. That means 75% of them are tall and 25% of them is dwarf. And out of 100, 3, that means 75. Out of 101, that means 25. Got it? So he did that experiment, and I hope you understood what he got from this experiment. He understood that in F1 generation, no parents, okay, no plants showed the recessive parental type in F1 generation, but in F2 generation, he saw that yes, the recessive parent, which was this, my dear children, it was still visible to him. Now, going ahead, all about Mendel's law of inheritance. So we studied about the experiment. Now, want to know the findings that he did, that he found. So Mendel proposed that something is stably passed down from parent to offspring through gametes over generation. He called these entities as what? Factors. He got to understand that something was passing from parents to the progeny out there. That was what? Capital T, small t. So all of them are what, my dear children? They are factors, which you now call as what? Genes. Now, genes are units of inheritance. Genes carry information required to express specific traits in an organism. So in the DNA, you have genes. These genes are nothing but segments that would be coding for some proteins, and these proteins are something that decides our character, which is why we say that genes are something that is responsible for a specific trait, because they are responsible for production of some proteins out there. But Mendel was not knowing about genes and all, so he used the term factors out there. Now, alleles are different forms of the same gene that code for a pair of contrasting traits. Ma'am, I didn't understand. So over here, he spoke about factors, that factors are passing down and things like that. Now, when you talk about the pair of the factors can be different like this. Okay, so these things are known as what? Alleles. Alleles are different forms of the same gene, or what? Alleles are different forms of the factors that you say. And this is a pair of contrasting traits. Okay, in the pair of contrasting, you see them, both of them. Now, he proposed that in a pair of dissimilar factors, that is this, one dominates the other. One dominates the other and hence is called the dominant factor. So this one is the dominant factor, whereas the other one will be what? The recessive factor. He also said during meiosis, alleles segregate and only one is passed to each gamete. He said that whenever meiosis is going to happen, this is from one parent. Now, when the gametes are going to be formed, only one of them will be going to one gamete. Only one of them will be going to one gamete. Both of them cannot be in a gamete. So, this is what? Segregation. So, now I am going to sum up all of them and I am going to give you two laws out of it. So, first he said there are factors and then he said that factors occurs in pairs, my dear children, which you call as alleles, and one dominates and the other one is not dominant. It is known as recessive. Then he spoke that whenever you want to form gametes, they segregate. They undergo meiosis and they segregate. They undergo meiosis as in they become half. Okay, and they move to each gametes out there. Now, according to this, he put down two laws. The first law is law of dominance and the second law is law of segregation. So, law of dominance says that characters are controlled by discrete units known as factors. That factors occurs in pairs and in a pair of dissimilar factor, one would be dominating over the other. The law of dominance is used to explain the expression of only one parental character in the monohybrid cross. We studied the monohybrid cross. What do you mean by monohybrid cross? The cross where only one character is studied, that is height. So, in that we saw that all the plants which were like this in F1 generation, they were showing only one character. So, how do we explain it? By the law of dominance, because this particular allele which is there, it is the dominant one. Okay, so that is what is said. Law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in F1 and the expression of both of them in F2. Now, in F2, both of them are getting expressed, my dear children, okay, because of the same reason. It also explains the proportion 3:1 obtained at the F2 generation. So, you need to know the ratios. They are super important. Now, the second law which is there, it is law of segregation, which says that alleles do not blend, both reappear unchanged in F2. So, if at all there was some blending, okay, my dear children, if at all dwarf and tall blended, my dear children, you would not have saw that in the F2 generation, but you clearly saw that. That means alleles which are there, capital T, small t, they are not blending with each other. During gamete formation, allele pairs separate, so each gamete gets only one allele. I told you, when this is happening, only one is gonna go to each gamete out there. Now, homozygous parents produce identical gametes while heterozygous parents produce two type of gametes. Homozygous parents as in like this, they are going to produce same type of gametes. Whatever you do, 100% of them will be capital T only. But if you talk about heterozygotes, at least some of them capital T and some of them small t. That means 50% of them small t. So, law of segregation says that whenever gamete is being formed, my dear children, the alleles are gonna separate and only one is going to go to each gamete, as simple as that. Okay. Now, going ahead with the next thing, is there any easy way to cross? So, we have seen cross like this, like capital T, small t, this and this like that. But is there any easy way? So, someone is saying, yes bro, and my name is Reginald C. Punnett. Who was Punnett? He was a British, please remember, British geneticist, and he was the one who gave down something which was known as Punnett square. So, whenever you want to cross something like this, what do? One side you put like this, capital T, and the other side you put small t, and then you cross this and this, capital T, small t, this and this, capital T, small t, this and this, capital T, small t, this and this, capital T, small t. This is how you do the math. Okay. So, this was put down by Reginald C. Punnett, who was a British geneticist. Now, going ahead, ma'am, I have a doubt. How did Mendel get to know if the plant is tall, like this or this? So, I know, okay, I am seeing that the plant is tall, but how do I get to understand whether it is homozygous tall, because both of them are same, or it is heterozygous tall? For that, what do I need to do? This is a very important question concept, my dear children. For that, I need to do something that is known as test cross. What exactly happens in the test cross, my dear children? You are going to basically cross the organism which has the dominant phenotype. The organism which has the dominant phenotype. It can be this or it can be this. With what? With the recessive parent, that means small t, small t. Okay, so you're going back and you're crossing specifically with the recessive parent. What's the use of it, ma'am? Let's see. So over here you have small t, and over here let's take this first. Okay, sorry, just a second. Okay, so over here I am going to take this first. So capital T, I'm going to cross it. So this and this, capital T small t, this and this, capital T small t, this and this, capital T small t, this and this, capital T small t. All of them are capital T small t. That means all of them are going to be what? Tall, my dear children. But now if I try to cross this with this, let's see what happens there. So over here I'm crossing, okay, capital T small t and small t small t. This and this, capital T small t, this and this, capital T small t, this and this, small t small t, this and this, small t small t. Now you tell me how many of them will be tall? This is tall, ma'am, this is tall, ma'am. So 2 of them are tall. What about them? They are dwarf. There is no dominant character. So 2 of them will be tall and 2 of them will be dwarf. So 2:2 ratio you get. That means 1:1 ratio you are getting. That means it was not tall like this. Here, the plants which appear to be tall, they were heterozygous. So that is the use of test cross. Okay, I hope you got now. Here the results are different when you cross both of them. So in a typical test cross, an organism showing a dominant phenotype and whose genotype is yet to be determined, we do not know that, is crossed with a recessive parent. I am crossing it with a recessive parent instead of self-crossing, and this is known as test cross. Now ma'am, this example is given in your NCERT where you have a violet flower. So violet can be like this, WW, capital W, or it can be capital W, small w. The cross was done and the same thing was observed out there. So now test cross versus back cross, what is the difference between that? In test cross, what you're doing? You're anyway crossing with a recessive parent out there. You're going back and you're crossing with a parent, right? Once the parent are there, then only children can be born. So you basically are going back and crossing it. So this is back cross. But when you are specifically doing it with the recessive parent, you call this as test cross. So crossing an individual showing a dominant phenotype with its recessive parent is test cross. And now crossing an F1 hybrid with either of its parent, it can be recessive or dominant. That is known as back cross. So can I technically say that test cross is a type of back cross? Yes, ma'am. You can definitely say that. Okay. Test cross is a type of back cross. Here I am specifically crossing with a recessive parent. That's the only thing out there. Cool. Got it? Thumbs up. Okay? Chalo. My dear children, if at any point you have some doubts, put it down in the comment section. If at any point you feel that this lecture is going super awesome, you have to tell me that also through the comment section given below. Okay? Chalo. Now all about the exceptions of Mendel's law. So Mendel said all of this thing, the law of dominance, one will be dominant, other will be recessive, and then he said that segregation, in segregation, my dear children, only one allele will be going in each gametes out there. So are there some exceptions to what Mendel said? So answer is yes, definitely there are. And one such exception is incomplete dominance, which you see in snapdragon or Antirrhinum species. What do you mean by incomplete dominance? That means here neither allele is completely dominant over the other one. The offspring shows a blending of traits which was not at all visible. That means if you're talking about capital, you're expecting it. R is red, okay, and small r is white, suppose. You're expecting this to be red only, and you are expecting this to be what? White, my dear children. Now when you form the gametes, capital R is over here, okay, small r is over here. When you cross them, capital R, small r, capital R, small r, capital R, small r, capital R, small r. All of them are capital R, small r. Capital R is dominant over small r, and which is why it should be red in color, but no, it will be pink in color, and this is what you mean by incomplete dominance. They are not completely dominant over each other, okay, and that's why you see a blending out there. Where do you see? In snapdragon. Also remember, Antirrhinum is the same thing out there. And now this was the experiment that he did. So as I told you right now, red and white he crossed. So over here, since all of them are capital R gamete only, so that's why they put it like this. So all of them would be pink in color. Now suppose I'm very curious, I want to cross them again. I'm going to cross the progenies among themselves to finding the F2 generation. Let's see what happens there. So capital R, small r here, capital R, small r over here. This and this comes together here, capital R, that means this will be red. This and this comes together, capital R, small r, that means it will be pink. Again, this and this comes together, capital R, small r, it will be pink. This and this comes together, small r, it will be what, my dear children? It will be what? It will be white in color. So if you talk about the phenotypic ratio, one is red, one is white, two of them are pink in color. 1:2:1 is the ratio. If you talk about the genotypic ratio, 1 capital R, 1 small r, 2 capital R, small r. So 1:2:1 again is the phenotypic ratio, and that is exactly given over here. Phenotypic and genotypic ratio remains the same in F2 generation in incomplete dominant plants, my dear children. So 1 red, 2 pink, 1 white. That is the phenotypic, physical appearance. Genotypic, capital R, small r, 1:2:1. Cool. So this is one such exception of what Mendel's cross. He says, Corin was the one who did the experiment. I said that no, there is something that is known as incomplete dominance also, where the alleles are not completely dominant over each other. Now comes the next thing, that is multiple allelism. So you know, when Mendel was doing his experiment on the monohybrid cross, that is for height, he was saying there is capital T and there is small t. These two are the alleles of the gene that codes for height. Now, when people started studying more, they got to understand that no, there can be multiple alleles also. Okay, there can be what? Multiple alleles. So, in the case of blood group, my dear children, you have 3 different alleles which are responsible for our blood groups. One is IA, second is IB, and the third one is IO. Okay, when there are multiple alleles governing a character, it is known as multiple allelism. When there are multiple things governing a character, that is your blood group, it is known as multiple allelism. And how does it work over here? Let's see that. So the plasma membrane of the red blood cells, my dear children, if you see, they have sugar polymers that protrude out of them. Okay, at the top, at the the surface level, they protrude out from the surface. And what kind of sugar should be protruding out? That is controlled by the gene. And the gene has 3 alleles, IA, IB, I. And now I'll come to basics of it. So this chart is given in your NCERT. Before going ahead with that, I'll try to explain you in my way. So for A blood group, B blood group, AB blood group and O blood group, what do you need? For A blood group, I should get one IA from one parent, another IA from another parent. In this case, I'll have A blood group. Or I can have one IA from one parent and one IO from another parent. Ma'am, if it's showing A blood group, does that mean that A is dominant over O? The answer is yes, it is. So you got to understand that O is recessive over here. Now going ahead, if I want B blood group, I should get one IB from one parent, another IB from another parent, or again one IB from one parent and one IO from another parent. Ma'am, again B is dominant over O, that means O is again recessive? Yes, you are absolutely correct. Now if I want AB blood group, my dear children, how can I give that? So one parent should give IA, another parent should give IB. In this case, they will have AB blood group. Ma'am, how can that be possible? One should be dominant? No, they are not dominant. And this is an example of codominance. Both of them are equally dominant, my dear children. There is only not one which is dominant. They are both of them are equally dominant. That is why it is AB blood group. This is again an exception. So multiple allelism is shown by blood group and also codominance is shown by blood group, and both of them are exceptions to Mendel's law of inheritance. Now going ahead, if we talk about O, ma'am, so B is dominant, OA is anyway dominant. So if I have to have O blood group, both the alleles should be the same. In homozygous recessive condition, I can show this. Okay, got it. So how many genotypes are possible over here in blood group? 1, 2, 3, 4, 5, 6. 6 genotypes are possible in blood groups, my dear children. And how many alleles do you have? You have 3 alleles out there. Okay, in this case, both A and B sugar would be protruding outside. That is why it is codominant. I hope you got it. Now, you can see this particular picture. Where is it? Here they have given the same thing out there. Okay, they are saying allele from one parent, allele from A here, then IA, blood group will be A. Okay, I'll take one random thing. IA and small I, what do you mean by that? Small I means O. So IA and O means it will be IAI, small I. That means it will be A blood group because A is dominant over O. So that is the thing. Okay, so codominance is the third exception. So multiple allelism, codominance and incomplete dominance in Snapdragon. I hope you got it. So over here what is happening? The plasma membrane of the red blood cells would be having sugar polymers, and what sugar polymers are decided by the gene, and that gene has these many alleles. Now going ahead to the next thing, polygyny. This is also something unique. This is also an exception, but not exactly given in your NCERT just there. It's given elsewhere, but I'm telling you it's an exception. Why an exception? So according to Mendel, he said that one gene is there that codes for one character. Agreed? But over here in polygyny, my dear children, multiple genes are there responsible for a character. For example, if you talk about your skin color, Okay, it is actually controlled by so many genes. Suppose over here capital A, capital B, capital C, capital C. Okay, so this gene are all controlling your skin color. So over here the genotype is capital A, capital B, capital C, capital C. Now suppose over here I replace capital A with small a. So now the whole thing is changed. Capital A, small a, capital B, capital C, capital C. Now if again I change this, I get small b. So now capital A, small a, capital B, small b, capital C, capital C. Whenever you see the difference is coming, a recessive gene is joining, my dear children, you will see different shades of color. This is the darkest skin tone, and now you are reducing the skin tone levels. At the end, if all of them are like this, recessive, this is the lightest skin tone available. So don't you think multiple genes are responsible for one character out there, and the same goes with the height as well. That is why we do not just have tall and dwarf, we have so many different height structures out there, because there are so many multiple genes which are responsible for this character. Okay, will you forget it? I hope you do not. And always remember, this will be having the lighter skin tone. So traits which are controlled by multiple genes are known as polygenic traits. Example, skin color and human height. Each dominant allele, capital A, B, C, adds to the trait, while recessive allele reduces it. Okay, it reduces the whole thing. Now, more dominant alleles result in darker skin, while more recessive allele leads to what? Lighter skin. Which is why we have so much variation. Okay, why we have so much difference in our skin tones, my dear children? Because multiple genes are being associated. So, polygeny is polygeny. Do not get confused with pleiotropy and polygyny. Polygyny means multiple gene. Poly means multiple. Gene means, you know, right? Multiple genes are responsible for one character. Now the fifth accession is pleiotropy. What is pleiotropy? So we again studied that one gene is there, ma'am. One gene is there and it is responsible for one character. No, over here one gene is actually responsible for two character, or basically multiple character. This is an example. Okay, this is an example that you see in our human body when a condition arises which is known as phenylketonuria. So phenylketonuria is a disorder in which what happens, the gene that codes for an enzyme phenylalanine hydroxylase is not formed. Okay, this enzyme is not formed. So then what happens? In our body, whenever we take in food, we get some amino acids. Okay, and one such amino acid is phenylalanine. This phenylalanine usually gets converted to something that is known as tyrosine, which is another amino acid, when phenylalanine hydroxylase is present. But now since this gene is not working well or mutated, my dear children, this is not happening, which is why what happens? Phenylalanine gets accumulated in the form of phenylpyruvic acid. And this phenylpyruvic acid, when it gets accumulated in our body, it causes mental retardation. And also, it is not absorbed by a body. It is not reabsorbed by a body. It is usually given down through urine. And when it passes through urine, it gives a very foul smell, a mousy smell. So always remember, in this condition, when phenylalanine hydroxylase is not being formed, what happens, my dear children? It forms phenylpyruvic acid, and this causes mental retardation. So ma'am, okay, one gene, one defect. No. The second effect, the second character which is being governed over here is that since tyrosine is not being formed, my dear children, what will happen? Tyrosine is something that is required for formation of melanin. Melanin is responsible for our skin or hair pigmentation as well. So if melanin is not there, what will happen? You will have a reduced skin and hair pigmentation. Got it? So when a single gene influences different metabolic pathways or phenotypes, when a single gene is responsible for different metabolic pathways or phenotypes, physical appearance, this is known as pleiotropy. Phenylketonuria is an example caused by mutation in the phenylalanine hydroxylase gene. The phenylalanine hydroxylase enzyme converts phenylalanine into tyrosine, and its absence, phenylalanine accumulates in the form of phenylpyruvic acid, causing what? Mental retardation. The second character it hampers is reduction in hair and skin pigmentation due to low tyrosine level. Okay. So fifth exception also you got it. So incomplete dominance, codominance, multiple allelism, polygyny and pleiotropy, all of them are exceptions. Now there is one example given in your NCERT, okay, that is about this particular thing. Capital B, whenever is there is large starch grains which are being produced, and this also signifies that they are round. Okay, so it is responsible for two character. Isn't it responsible for two character? This particular gene set, it is responsible for the shape and also for the, the size. large starch grain and also seeds are round. Now small b, small starch grain and the seed becomes what? Wrinkled, my dear children. But now this is also an example of incomplete dominance. Why? Because when capital B small b comes, this is not completely dominant, which is why you get medium starch grains. Otherwise you should be getting large starch grains, right? So this is an example of incomplete dominance as well. Now seeds are round but the starch is what? Intermediate. The starch which is where is there, it is intermediate. What does this show? For seed shape, B is dominant. But for starch grain size, it is incomplete dominance, and this is an example of pleiotropy as well, because one gene set is responsible for multiple things. One is shape and the other one is size. Okay, so this won't be asked, but as it was there in the NCERT, I just discussed what if they change their mind and ask someday. So yes. So with that we complete all the exceptions which has there, okay, and we covered what the Mendel's monohybrid cross, and now we are going to go ahead with dihybrid experiment. Dihybrid means you are studying two characters at once, my dear children. So over here what exactly happens? Junior Mendels, I hope you are ready for the experiment. So over here I am taking round and yellow. Just a second. Okay. I am taking round and yellow plant, and over here wrinkled and green plant. So round is a character, yellow is a character. Two things have been taken up, round is the shape and yellow is the color. So that is why I call this dihybrid cross, because now I am going to cross them. So here the dominant ones I have taken capital R, here the dominant is capital Y. Similarly small r, small y, small y. I have taken that, and now whatever gametes are going to form, they are going to be formed like this. Ma'am, I didn't understand a word. So listen to me carefully. I will make you understand this. It's very simple, okay? So, capital R, capital Y. Small r, small y. This is what? This is round and this is yellow. This is what? This is wrinkled and this is green, right? Now over here, if you talk about the chromosome, it looks like this. So capital R, capital Y. Similarly, small r, small y. Okay, got it? Now from this pair only one can come in. Suppose capital R, from this pair only one can go. Either way, whatever it goes, it will be capital R, capital Y. Similarly, from this pair small r, this pair small y. Right, whatever you do, these type of gametes are going to be found. Right, from a pair only one can go. Here also from a pair only one can go. Now this gamete and this gamete fuses, my dear, and what will happen? Again, capital R, small r, capital Y, small y. So it will be what? Capital R, capital Y, small y. This is the progenies or F1 generations genotype. And what is the phenotype? Capital R is dominant over this, so it will be round, and capital Y is dominant over this, so it will be yellow. Got it? So we got this. Mendel was not happy. He wanted to do again an experiment. He wanted to cross something like this. So he crossed first initially the parents, and now he is crossing them, my dear children. So in the first generation he saw the dominant parental character, but now let's see what happens in the second generation. Okay, so now when you are crossing this, how does this appear to you? So capital R, small r, capital Y, small y. Capital R, small r, capital Y, small y. Okay, now here the condition is something like this: capital R, small r, capital Y, small y. Similarly, capital R, small r, capital Y, small y. So from here only one guy, suppose this came, from here one guy, this came, so this is a gamete. From here again capital R came, from here small y came, this is a gamete. From here small r came, from here capital Y came, this is a gamete. From here small r came, from here small y came, this is again a gamete. So 4 type of gametes are being formed this time. Similarly over here also you will get 4 type of gametes being formed. So I am writing the 4 here, okay. So capital R, capital Y, capital R, small r, capital Y, and small r, small y. Now if you want to cross, you won't do zigzag cross like that. You will make the Punnett square and cross it. This and this crosses, you get capital R, capital Y, capital Y. This and this again capital R, capital Y, small y. You cross in this way, you will get 16 progenies out there. And what did he find? He found that, ma'am, 9 of them, the most number of them, were round and yellow, just like how it was there in the dominant parent. Only 1 of them out of 16 was showing the recessive parental character, that is wrinkled and green. Only 1 of them. 3 of them are something new. So round was with yellow, no? until now, but here this time round will be with green. 3 of them will be round and green. And the next 3 will be what? Wrinkled was with yellow. The next 3 will be wrinkled was with green, and this time wrinkled will be with yellow. These are the new combinations that you got. So out of 16, 6 of them were new combination. 9 of them were the dominant thing. 1 of them were the recessive parental type. So what did he get to understand from this? He put down the next law, that is law of independent assortment. According to it, what does that mean? Listen, my dear children, we studied law of segregation. According to it, whenever you are going to make the gametes, from this pair only one will be coming in. That's what you said. Now, law of independent assortment means whenever you are talking about one character and second character, the alleles for this character are going to separate independently of the alleles of the another character. Didn't understand? I'll tell you. So, it is not like capital R will only go with capital Y. Capital R can go with small y also. From this pair anyone can come, from this pair anyone can come. They are not going to assort dependently. They are going to assort independently, which is why you see new combinations. Otherwise, you would not have seen newer combination. So, that is law of independent assortment. So, if at all you are given this equation or ratio, my dear children, and you are asked what law does it shows? It shows the law of independent assortment. That is why you are getting new combinations. If at all it was same combination, you will get the same type of organisms only. No new type of organisms are there. Okay. So, did Gregor Mendel find something unique out of it after doing this experiment? The answer is yes. The law of independent assortment, the third law, my dear children. And according to it, what does it say? It says that when two pairs of traits, one pair of trait, as in you are talking about this shape, another color, are combined in a hybrid, segregation of one pair of characters is independent of the other pair of character out there. So, round and yellow are not going to go hand in hand, my dear children. Round can go with green and yellow can go with wrinkled as well. Okay, you need to know that. That is law of independent assortment, and that is what he found out after doing his dihybrid cross. Now, but what went wrong? Okay, he did his experiments for 7 years. Do you remember from what year to what year? I told in the initial part of the session, but if at all you remember, put it down in the comment section right now. Okay, so what went wrong even after doing these many experiments, my dear children? First thing, in his time, communication was so difficult, which is why his work was not widely known. Second, his idea of discrete factors on no blending, my dear children, was not accepted because you clearly saw in incomplete dominance, there were blending that was observed. Using mathematics in biology was unusual and not accepted. So, initially we praised him for that only, using mathematics. But towards the end, there were so many people who said that no, I don't get the logic. Why you need to use mathematics in biology? And the fourth thing, he had no physical proof of what genes were or how they looked. And that was a big trouble. Why? Because he didn't have a microscope back then, my dear children. He didn't have the clear-cut things out there. So, these were the 4 reasons why his things were not actually being studied well. So, communication was difficult, his idea of discrete factors and blending was not accepted, of no blending was not accepted, and using mathematics was not accepted, physical proof was absent. That is the reason. But then came a twist. What was the twist, my dear children? After 37 years, imagine, 3 people came in, okay, in 1900, Karl Correns, Erich von Schemack and Hugo de Vries. Very important, the year also very important, 1900, 1900, okay, 1900. So, Karl Correns, Schemack and de Vries, the 3 of them, they came together. They were better microscopes, revealed that chromosomes in the nucleus which doubled and split during cell division. So, by this time you had they had the microscopes, and they also got to understand that yes, there were chromosomes out there which were separating from each other during meiosis. Okay, and they backed up what Gregor said. Okay, he said that yes, something somewhere is actually correct, and they were the people who rediscovered Correns, Schemack and de Vries. Now, after 1900, 2 years later, 2 more people came in, and it was what? Sutton and Theodore. Okay, Sutton and Theodore Boveri, who came together in 1902, and they said that behavior of chromosomes was parallel to the behavior of genes and used chromosomal movements, or basically they said something more important which I can talk in the next slide, and that is this chromosomal segregation with Mendelian principles and called, okay, Pairing and separation of a pair of chromosomes would lead to segregation of a pair of factors that they carried. Didn't get it? I'll tell you. So they said that, okay, chromosomes were there, I am able to study it and I am able to see the factors. So whenever chromosomes moved away from each other, I was able to understand that, okay, the factors are also moving away. So that means probably these factors are present on the chromosome itself, and that is exactly what Mendel said. He didn't use a term chromosome or anything else, but he said that there were some factors which were segregating and going to each gametes out there. So these factors were there on what, my dear children? On the chromosomes. On the chromosomes, and that is what these people said. Who are these people? Walter Sutton and Theodore Boveri in the year 1902. They said that pairing and separation, pairing or separation of a pair of chromosomes would lead to the segregation of pair of factors also that they carried. So Sutton was the person who united the knowledge of chromosomal segregation with Mendelian principles, Mendel studies, and called it chromosomal theory of inheritance. Actually, it was put down by both, but in your NCERT, one very important thing is given, that is, they mentioned Sutton predominantly. Okay, they said that Sutton also gave the chromosomal theory of inheritance, and that is this. Now obviously they said this, but someone has to prove all this thing. Who was that someone? It was Thomas Hunt Morgan. Okay, it was Thomas Hunt Morgan who did an experiment to prove the chromosomal theory of inheritance, that yes, chromosomes segregate and also the factors would be there, they also segregating. So for that, he used a very important organism that is known as Drosophila melanogaster, or which you call as fruit fly, my dear children. But the reason why it is used is very important, because again you can get questions out of it. So they can be grown on simple media, first thing. Their life cycle is very short, only 2 weeks. So after 2 weeks you can see whether, okay, what changes are being coming up. High progeny count will be there, that means so many babies would be formed out there. Clear male and female distinction is there. The male is the one that is shorter and the female is the one that is larger in size. Visible hereditary variations were there under low power microscope. So under low power microscope also he could see the variations out there. That is the reason why why he used fruit flies or Drosophila melanogaster. I hope it's crystal clear. The 4 reasons or the 5 reasons out there, very, very important, write it down. Okay. Now, did he find something else? Okay, he did his experiment and all, but did he find something else? The answer is yes. He found 2 things. One is linkage and the other one is recombination. So initially I thought I'll show it to you like that, but okay, I can explain here itself so that it can save a lot of your time. Okay, so over here, suppose you have 2 chromosomes like this. Okay. Okay. Now, in the case of chromosome, they have a term that is known as crossing over, where a part of the chromosome, they move like this, they swap. So whenever the swapping thing is happening, suppose, my dear children, what will happen? Here, small a and small b will still be together. Capital A and capital V will still be together, even though they change the position, but they are still together. On the other hand, if you see over here, my dear children, suppose swapping is happening here. So capital A comes this side, but small b remains here. Small a goes that side, but capital B remains over here. So here you see what? A new combination arising. But here do you see that combination? Newer combination arising? Very less. No, right? The lower part remains the same. Only the upper part would be having some changes if at all there is any. You get it? So over here, can I say the chances of newer combination, or what do you say, recombination, okay, that would be comparatively less. Why, ma'am? Because the genes over here, they are what? They are tightly linked to each other. They are very close to each other. They have very less distance between each of them. Whereas when you talk about this, here you see a newer combination being formed, capital A with small a with capital B, very new combination. So can I say here the chance of recombination is more? Why? Because the genes which are here are not tightly linked, but they are what? Loosely linked. Loosely linked genes means they have longer distance between them. Tightly linked genes means they have shorter distance between them. Don't try to mug it up. Imagine and study. If at all there are two close friends, do you think someone else can enter and form a new friendship out there? No, they're very close already. So less recombination. If they are far away from each other, friends are not really close, another friend can come in, there can be new combination as well. So this terms linkage and recombination was given down by Thomas Hunt Morgan, my dear children. And along with that, so over here I have written the same thing, tightly linked, loosely linked genes, so I don't have to explain it to you once again. And among that, after that, my dear children, he had a student and the student rocked and a teacher got shocked. Why do I say so? Who was the student? What is his name? His name is Alfred Sturtevant. His name is Alfred Sturtevant. You might have seen it in your NCERT, Sturtevant. What did he exactly do? So he actually discovered gene mapping. So Morgan said that yes, these genes are located probably here and here, which is why they are far away from each other, which is why recombination rate is also more. So with that information, Alfred Sturtevant came and said that okay, probably the genes might be located here and here. He was able to map the genes with the help of the recombination rate and everything out there. Now does it make sense? I hope it does. Okay, so this was said by Alfred Sturtevant. Cool, done. That is why I said students rocked and teachers shocked. Now over here in your NCERT you might have seen this, but I'll say do not invest too much of your time into it. I have explained it in the in some part video, I think part 4, okay, L4 of principles of inheritance in Sankalp. But I am just telling you, if at all they are giving you something like this, that is tightly linked genes. Okay, wait. Tightly linked genes very close to each other on chromosome, very low recombination. Example, white and yellow genes show 1.3% recombination. 1.3% recombination is very less recombination. That means whenever you are saying that something is showing 1.3, that means they are tightly linked. That means they do not have any space between them, or no much space between them. That's what I mean. Now in here it is said that white and miniature wing genes show 37.2% recombination. 37.2% recombination means too much amount of recombination, and if too much amount of recombination has to happen, that means the genes are located far away from each other. They are loosely linked. You get it? So that type of questions would be coming in. This percentage of recombination is there. What do you mean by that? That means it is very far from each other. They are loosely linked. So 50% is the maximum. So 37.2 is very close to 50. Okay. So you get to understand that. I hope that's clear. And 1 marker question, I have made you solve this question. So please don't worry. You can just see it. Now going ahead with the next thing, that is all about sex determination in the case of organisms out there. So there was a guy whose name was Henking, and he was studying the sex determination organism. He came to know that some sperms which were there, they carried the X chromosome, and some sperms out there, they didn't carry anything out. There is nothing, actually no chromosomes out there. So 50% was like this, 50% was like this. He kept on doing his experiment, and that's when he realized that there are different type of sex determination. And one such sex determination is what? XO type of sex determination, where what is exactly happening? Here the females will be having XX chromosome. That means they can produce X in the gamete or X in the gamete. Same thing. But in the case of males, either there can be X or there can be nothing, which is denoted by O. Now over here, suppose this and this came together, you get XX, the female. When this and this comes together, my dear children, you get XO, and this will be turning out to what? Male. So this type of sex determination, where the males are producing two different type of gametes, this is known as male heterogamety. Hetero as in different, gamete as in different gametes. This is what? Male heterogamety that is being shown over here in XO type of sex determination. Where do you see it, ma'am? In the case of insects. For example, grasshopper is one such example out there. XO type of sex determination is found in grasshopper. Okay, please remember it. And now next thing that we have is XY type of sex determination. How in human beings we have it, right? The same thing is there in Drosophila, that is fruit fly. So remember, T.H. Morgan and Drosophila are friends. Both of them have XX, XY. Do not think Drosophila is an insect, so it will have XO. No, here count them together, same. So over here also, if you see, the females are going to produce same type of gametes, XX only. But the males, X and Y, they are producing two different type of gametes. So again, this is an example of what? Male heterogamety. 50% of chances there a female will be born, 50% of chances there the male will be born. How, ma'am? So suppose this X comes together with this X. Okay, so you get XX. This X comes together, this Y. You get XY. This X comes together with this X, you get 2X, that is female again. This X and this Y, again you get a male. So 50% male, 50% female. A question is there. Parents had thrice girl child. What is the chance of a boy child being formed? 50% only, whatever you do. Okay, whatever you do, it will be 50%. And here it is an example of male heterogamety. Two examples of male heterogamety, XO type and XY type. Now the next type is ZW type of sex determination. Where do you see it? In birds. And this is the most different case out there, because this is where you see female heterogamety. Females are the one carrying Z and W. So one type of gametes carry Z, another type of gametes would be carrying W chromosomes out there. So Z and W. Okay. Now suppose over here males will be producing same type of gametes. So this is male homogamety. There is female heterogamety. Okay, so suppose this and this comes together, you get ZZ. Suppose this and this, you get ZW. Similarly, this and this comes together, ZZ. This and this comes together, ZW. So 50% chance female, 50% chance male. Always in sex determination, 50% chance will be there for male and 50% chance will be there for females as well. Got it? And this is an example of female heterogamety. I hope you do not forget it. It's there in birds. Female heterogamety, female produce two different type of gametes, ZW in the case of birds. Male heterogamety, males produce two different type of gametes, XY and XO. Perfect. Now there is next type of sex determination which is seen in honeybees, and this is haplodiploid sex determination. Haploid means half the number of chromosome. Diploid means the whole number of chromosome, 2n. So females, my dear children, they are generally diploid in nature. Just a second. They are diploid in nature. They have 2n number of chromosome, and whenever there is diploid, you have to undergo meiosis to form the gametes. You have to half them down. Okay, so you half it down, you get N and N over here, N over here, N over here. Similarly, in the case of males also, if you see, they are already haploid. They already have half only. So here 32 number of chromosomes were there. Here they only have 16. So can you make it half anymore? You do mitosis instead of meiosis. So here the number of chromosome is what? 16. So now what happens? Suppose this gamete and this gamete fuses. 16 plus 16 comes together 32. Will it be a male or female? It will be female because it is diploid. What about this one? This one, without undergoing fertilization, it will transform into male. This is not going to undergo fertilization, which is why the N remains to be 16 itself. There is no doubling, okay, because there is no fertilization happening, and this is known as parthenogenesis. So the males that will be formed, my dear children, they will be always having half the number of chromosomes. They are going to be haploid. But the females, definitely they are going to be diploid. Okay, so fertilization is happening, then female. If fertilization not happening, then male. Okay, and there is one statement given in NCERT, they do not have father and thus cannot have sons, but they have grandfather and can have grandsons. Why they do not have father? Do fertilization happening over here? No. But my dear children, this particular female might have had a father, then only we can get a female, right? So that is why it said they have grandfather and grandfathers have grandsons, they do not have sons. So fathers cannot have fathers, and they can have grandfathers for the same reason, and they can have grandsons also for the same reason. No father-son relation is there. That's what you need to remember. Okay. Now all about mutations. Okay. Let me breathe. What do you mean by mutation? Mutation is a phenomenon where, my dear children, DNA sequence is getting altered, and whenever the DNA sequence is getting altered, what will happen? There will be obviously some changes in the proteins that is being formed, and that whenever that happens, we say the phenotype is changed, the physical appearance gets changed. Similarly, it leads to change in the genotype. So both genetic makeup and physical physical structure is going to be changed whenever some mutation is going to happen. And mutation in your NCERT, two different types of mutations are being mentioned, which you need to remember. One is point mutation. Oh shit, just a second. Okay, one is point mutation, okay, and the other one is what? Frame shift mutation. What do you mean by point mutation? Point mutation means, for example, if you talk about this particular strand of RNA out here, okay, you know, our DNA, everything are made up of so many nucleotides. Okay, they are nucleotides. So over here you have GUG, or basically you can just consider you had GAG. Initially you had something that is known as GAG. These three things are known as a codon. A codon is the one that codes for an amino acid. It will be coding for what? An amino acid. So here this codon codes for amino acid, this codon again will code for an amino acid. All the amino acid will come together to form a protein. Suppose there is some change over here from GAG. Now this A is replaced. At one point, this A is replaced and it is converted to U. Do you think the same amino acid will be formed? No. So over here, initially glutamic acid was being formed, which is an amino acid, and now since this replacement has taken place, it is valine which is being formed, my dear children, which is why the whole protein which is being there, its structure would be totally different. So this is an example of point mutation which you see in sickle cell anemia. It's a disorder where you see what? Point mutation. Now frame shift mutation is also there, where suppose you are inserting something out there, okay, suppose you are inserting, so example over here I have 1, 2, 3, 4, 5, 6, 7, 8, 9. Now over here I am inserting one more thing. So obviously will it be 1, 2, 3 now? No. This new thing will be taking up the place. This new thing will be taking up the place. So there is an insertion, or whenever there is deletion, my dear children, that will also lead to what? Change in the frame. And whatever codons are there, that amino acid will be anyway formed. So even if there is a change in even a single nucleotide up there, my dear children, there will be a change in the amino acid that is being formed out there. So this is an example of frame shift mutation. Okay, I hope that's clear. And we will talk about sickle cell anemia way more when we come to that topic. Okay, now going ahead, talking about the pedigree analysis, all about the pedigree analysis. So what exactly happens over here? What exactly is your pedigree? So pedigree basically means analysis of traits in a several of generation of a family is known as pedigree analysis. You are analyzing a trait in each of the generations out there, and you need to know all of this thing. Whenever there is square, it means it's a male. Whenever there is circle, it means it's a female. Whenever it's a shape like this, that means sex is not specified. If you color all these things, that means they are affected individuals. If you show one line between the male and the female, that means mating is happening. If you show two lines, that means mating is happening between relatives. That is why they close. That is why two lines are there. Consanguineous mating. What do you call this? Consanguineous meeting. That is what you call it. Mating, sorry. And now over here, this male and this female have mated, and now they have two kids. One is female and the other one is male. Okay, now over here this male and female have mated, and now they have got affected child. Suppose over here they have mentioned like this, where they have made a shape and written 5, that means 5 children are being born, but the sex is not specified out there. And this one is the most important out of all of it. Okay, yes. Now what about disorders? This is the last topic, and with this we are going to end. So before I start ahead, my dear children, also one more very important thing. The things that causes mutation are known as mutagens. What do you call? Mutagens. For example, UV radiation, all of it are mutagens. So that's why we say do not have your mobile phone near you when you sleep. Now Mendelian disorders. So there are two types of disorders, as I told you. Mendelian, no, I didn't tell you. So Mendelian and chromosomal disorders. Mendelian disorders means, for example, okay, this chromosome is there, the gene is being mutated out there, which is why I have some problem, versus why I have some disorder. So they will be categorized under Mendelian disorder, when it is determined by alteration of mutation in the single gene out there. Now chromosomal disorders are the one which is determined by alteration in the structure or the number of chromosomes. For example, instead of having a pair of chromosome, I have one extra chromosome. Now I will have a disease. Instead of having a pair of chromosome, I do not have the pair itself. So that's also how I will have a disease. That is chromosomal disorder. Okay, clear? Now before I go ahead, I want to talk about autosomes and allosomes over here. So in our body we have 46 number of chromosomes, which we also call as 23 pairs. Right, my dear children? So 22 pair, my dear children, are autosomes. That means they do not have anything to do with our sex. Whereas 23rd pair, my dear children, they are known as allosomes or sex chromosomes. They can be either XX or it can be XY. They can be either XX or it can be XY. So if you are asked how many number of autosomes are there, we say 22 pair of autosomes are there, or 22 into 2, what's 44 autosomes are there in total. 45th and 46th chromosomes are sex chromosome. It can be either XX or it can be XY. Okay, they are also known as allosomes. Now, going ahead with the first disorder that we have, that is color blindness. And it is sex-linked recessive disorder. What do you mean by sex-linked recessive disorder? That means, my dear children, the gene which is affected over here is on the sex chromosome. Okay, and if I say that it is recessive disorder, that means, for example, if this gene is affected, In this condition, a person will be having the disorder. Suppose if it's like this, one gene is affected but one is normal, this will not show the effect. Why? Because there is a dominant one to overshadow the effect. Only in homozygous condition it will show the effect. You remember small t when it comes together, that's when you get dwarf. Otherwise, do you get dwarf anyways? No. So that is why he said it is what? Recessive disorder. Why it is not sex linked? Because these genes which are there, which you are talking about, which are defected, are on the sex chromosomes. Okay, or on the X chromosome out there. One example is color blindness. What exactly happens over here? Listen to me. The same thing is going to happen in the case of hemophilia as well. Okay, so over here, males have XY, females have XX condition, right? Suppose I have one X which is having mutation out there. There is another X which does not have it, which is why this particular female is just going to be a carrier. She is not going to have the disease. On the other hand, if the male is having one X which is which is having the damaged gene, do you think he will be a carrier? The answer is no. Why? Because it does not have the next X chromosome to overshadow the effect, which is why this guy will be anyway diseased. If at all he gets one X chromosome that is getting affected. Always remember this funda. Now, the situations I will give you. Suppose over here you have a condition like this. Now, this and this fuses, you get XX, XC. This and this fuses, you get XX, normal X. This and this fuses, you get XCY. And this and this fuses, you get XY. What do you mean by that? Here, a female is being formed and she is a carrier. Here, a normal female is being formed. And here, what do you call? Here, a man who is colorblind is being formed. Here, XY condition is there, normal, where no X is getting damaged. This will be a normal male. So, these are the conditions that you have. And you need to remember it. So, 25% of the females would be carrier, okay. Rest 25% will be normal, my dear children, or sorry, 50% of the female will be carrier, 50% of the females will be normal, 50% of the male will be carrier, colorblind, and 50% of them will be normal. If you're just talking about males, if you're just talking about females, then as I said, 50-50. Now, if you're talking about whole, then my dear children, 25% of them, out of 4 only 1 is a carrier female, 25% will be carrier female, 25% will be normal female, 25% will be normal male, and 25% will be colorblind male. So, in similar way, you have different things. For a female to be colorblind, so here you saw the carrier female is there, no colorblind female is there. For a female to be colorblind, what do you need to do? One chromosome comes from the father in the female and one chromosome comes from the mother. So, at least the mother should be a carrier and the father definitely should be what? Colorblind. Then only you can get this chromosome from him. And both the X chromosomes should be coming like this, then only the female will be colorblind, my dear children. So, the females to be colorblind, the chances are really less because of the same reason. Listen, females are usually carriers unless they inherit 2 defective copies, one from each parent. It occurs in about 8% of males and only 0.4% of females. This is very much important for you to remember. Okay, 8% of males and 0.4% of females. Why? Because the reason is this. Both the parents, at least she should be a carrier. At least. She can be a colorblind person also, but at least she should be a carrier and he should be colorblind. For a female to have this. If he is not colorblind, the male can definitely have this. As I said, if this and this comes together, the male will definitely have it. What exactly happens in color blindness, my dear children? So, over here there is defect in the red or the green cone cells leading to difficulty distinguishing about the red and the green color. And it's not just that, there are different types of color blindness as well. Now, a carrier mother has a 50% chance of passing to her son. A carrier mother, okay, a carrier mother has a 50% chance of passing it to the son. Why do I say so? Because, my dear children, son can be formed in 2 ways, this and this XY, this and this XCY. So, over here, out of 2, one son will be definitely having this. So, can I say 50%? Yes. So, that is what I said. A carrier mother has 50% chance of passing to her son. A daughter will be colorblind only if her father is affected and the mother is at least a carrier. I hope this is clear. This is the first disorder, that is the first sex-linked recessive disorder. And here you have so many conditions. If you can download the PDF, you will be seeing it. So, the rounded ones are the affected ones. Okay. You will be given different conditions and you need to tag them out then with the help of this. Now, Okay, the next sex-linked recessive disorder is what? Is this? Is what? It is hemophilia. Again, the same thing is happening over here, but here there is a twist. I'll tell you what's a twist. So suppose this is the one that is affected, XH, right? Now, this and this you get, what is this? She is a female carrier, okay? She is what? A female, normal female. He is what? He is a hemophilic man. He has hemophilia because there is no X other chromosome, and then there is XY. So if you have color blindness, my dear, you won't be able to see the color. But if you have hemophilia, it is not a disease that will let you live. Because what happens in hemophilia is that basically the gene that codes for a protein, a clotting protein, my dear children, it is getting affected. And if clotting is not happening, if you get a simple cut, you will keep on bleeding forever. You get it? So obviously, if a guy has hemophilia, he would not be able to survive. He would not be able to survive until long. And basically, he cannot marry to have kids. And if he is not mating with someone else who is at least a carrier, they cannot produce a female offspring that has hemophilia. So for females to get this disease, it's very, very rare. I hope you get it. Why? Because over here in color blindness, the males were there. Defective parents can be there, that's okay. But he here cannot survive that long that he can mate and produce kids. And if mating is not happening, defective, you need both defective parents, at least one carrier and one defective parent to get it. So you cannot take it. The possibility of a female to get it becomes very, very rare. The possibility of female becoming a hemophilic is extremely rare because mother of such a female has to be at least a carrier and the father should be hemophilic, unviable in the later stage of life. But it is unviable, that means they won't be surviving until that particular point of time. That is what is meant over here. Now, single protein that is a part of cascade of proteins involved in clotting of blood is affected over here. That is why simple cut will be resulting in non-stop bleeding. This is again the sex-linked recessive disorder. It's on the sex chromosome and it is recessive as well. Okay, so for a female to get it, she should have something like this, which is very rare. Cool. So we are done with the sex-linked ones, and now we are going to come and talk about autosome-linked recessive disorders. So autosome means they are on these genes, whichever is getting affected, it's on the autosomes. Again, you need a pair of them, defected one, to get that particular disease. Okay. So what exactly happens in sickle cell anemia? It's caused by the mutation in beta globin gene. So over here, listen, you know hemoglobin is the one that carries oxygen out there. So hemoglobin is made up of two things, alpha globin and beta globin. Okay. Now the beta globin chain is produced with the help of beta globin gene. Now suppose there is a mutation in this particular gene itself. As I told you earlier, suppose their codon was like this, GAG. But the point mutation is happening and it converts to GUG. Here this was coding for glutamic acid, which is one amino acid, and now here it is coding for valine, which is another amino acid. Since this valine is there, what will happen? The whole beta globin chain structure is changed now. It is produced, but it is changed. The quality, not quantity, quality is getting reduced over here. Now, the mutant hemoglobin molecule undergoes polymerization under low oxygen tension. Listen, since this beta globin is not that great, it won't be able to capture oxygen also, and within this particular time, there will be some changes in the hemoglobin structure, which will later lead to what? Change in the structure of the RBC. The RBC, which was usually biconcave in shape, will now turn towards sickle shape. And if it's now sickle shaped, what will happen? Can it transport oxygen? Do you think so? No. And not just that, it will help, it will clot, what do you call it? It will not clot, what do you say? It causes obstruction in the blood flow because something like this is there. When it was biconcave, it was easily moving, but now the shape of it has totally changed. You need to understand that now. And how do you remember this mutation? Remember, A comes before U, so it was A first and then it was replaced by U. Similarly, G comes before V, so glutamic acid was there first and then it was replaced by valine. Now, HBS disease individual. So over here, one important thing that you need to remember is that I was talking about the alleles, right? So suppose over here, HBA is there. This is normal allele which is there. So here the hemoglobin will be produced normal. Suppose one is HBA and the other one is HBS. Here, this person is a carrier. This person has the mutant allele. He or she can pass it to the next organism. Okay. Now, suppose it is like this. Both are HBS, HBS. That's when we say that a person has sickle cell anemia. Remember, this is not sex linked. Any guys or girls can have it. Okay, so if both the parents are at least the carriers. I am not saying both the parents have it, because again both the parents cannot have it. Why? Because they would be dying off. If they have the disease, they would be not surviving enough to produce offsprings. Okay, so at least suppose if they are carriers out there, suppose the condition is like this. HBA and HBS. Similarly, HBA and HBS. This S and this S, by chance it comes together in some offspring out there, my dear children, that person would be having the disease. That is why it is said if both parents are carriers, the child will also have it. Remember, this is an autosome linked recessive disorder, that is sickle cell anemia, and it is a qualitative blood disorder. Quality is getting problematic over here. It was GAG, GUG, glutamic acid, then valine. It's happening in the 6th codon, 6th position. So you know, when you talk about an RNA, which is responsible for forming the proteins out there, they have multiple codons, 1, 2, 3, 4. So on the 6th position, whatever codon is there, that is this, that's where the change is happening. Please remember that also. And where do you find beta globin chain? On which chromosome? How many chromosomes do we have? We have 23 pair of chromosomes, ma'am. So basically on chromosome number 11th, on the 11th pair of chromosome, my dear children, you will be having the beta globin gene. Please remember, 11th chromosome. Okay, and now with the same thing, we'll go ahead, not with this, I'll go ahead with the next disease, that is thalassemia. So thalassemia is more or less, what do you call, coming in the same category. One, because both of them are autosome linked recessive disorder. Second, both of them are blood disorders. But here there is a change. This disorder is a quantitative disorder. And what do you mean by quantitative disorder? What do you mean by here the quantity is getting disrupted, my dear children. You need to understand this. So over here we know alpha globin gene is there, beta globin chain is there, both of them forms hemoglobin. Suppose there is no much beta globin chain being produced. Do you think hemoglobin will be produced enough? No. If no much alpha globin gene is being produced, do you think much hemoglobin is going to be produced? No. So that is happening over here. Thalassemia is a case where a mutation or deletion reduces synthesis of either or alpha or beta globin chains of hemoglobin, leading to abnormal hemoglobin and anemia. There is very low level of hemoglobin, which is why it leads to anemia. In alpha thalassemia, what happens? It affects the alpha globin production caused by mutation in the HBA1, HBA2 gene of chromosome 16. So this is chromosome 16. There is HBA1 gene over here and here, alleles are there, and then HBA2 genes are also there, here and here. Suppose one gene is not being mutated, one gene is being mutated, still alpha globin gene is being produced but not much. Suppose both of them are being mutated, still alpha globin gene is being produced but not too much. If this is also getting mutated, still alpha globin gene is being produced but not too much. If this is also getting mutated, no alpha globin gene is being produced, which is why there is no hemoglobin also being produced out there. But either way, whenever you say you are a carrier also, you have very less amount of alpha globin chain, which is why you will be mostly anemic only. But if you do not have it at all, can you survive? No. Okay. So where is HBA1 gene and HBA2 gene? It's on chromosome number 16. There are two genes. I'm not saying two alleles. Two genes. This is one gene. I'll show you the picture. This is one gene. This is another gene. Both of them have two alleles of their own. Now this can be affected, that person would be a carrier. If both of them are affected, you have alpha thalassemia minor condition. If 1, 2, 3 alleles are affected, you have moderate to severe thalassemia. If all of them are affected, my dear children, you are a major patient of thalassemia. You cannot survive. You get it after some point of time. This is about alpha thalassemia. This is where on the chromosome number 16, I always say 6 looks like half B. Suppose I remove the B over here, it looks like 6. So people usually go with 6 with beta globin chain. No. 16 is ulta. It's for alpha globin chain. 11th chromosome is for beta globin chain. Okay, now if we talk about beta globin chain, there is only one gene, HBB. HBB gene, allele over here, allele over here. Suppose this is getting mutated, still there is someone. If both of them are getting mutated, my dear children, what will happen? You have beta thalassemia, you have a beta thalassemia major patient. So I hope you get to understand, it's a quantitative disorder. It's the quantity that is getting disrupted out over here. If you have enough, you still survive. If you do not, you do not survive. Okay, and it is usually being asked. Difference between sickle cell anemia and thalassemia, and I'll be discussing in the 2 and 5 marker questions as well. Okay, got it? So this is also autosome linked recessive disorder, and now you have autosome linked recessive disorder, one more, that is phenylketonuria. I already explained you. Okay, I already explained what happens in phenylketonuria. Phenylalanine hydroxylase is not being produced, which is why phenylalanine is accumulated as phenylpyruvic acid. This causes mental retardation, and also since this is not reabsorbed by a body, it goes out through the urine and it smells so bad. Okay, and since tyrosine is not being formed, melanin is not going to be formed, and hence there would be hypopigmentation. So my dear children, in total you studied 2 sex-linked recessive disorder. One is hemophilia, second is color blindness, and 3 autosome linked recessive disorder. What are those? Thalassemia, sickle cell anemia, and also phenylketonuria. And in total 3 blood disorders are there. One is hemophilia, but it is sex linked. 2 of them are autosome linked, and that is what? Sickle cell anemia and thalassemia. Okay. Now, in your NCERT, you have something that is known as pedigree. So, which one is quantitative and which one is qualitative? Can you tell me? This is your homework number 1 question. Now, you know, you can tell me. Not hemophilia, it is thalassemia. Okay. Okay. Now, over here, I need you to understand how does this particular thing work. That is what? The pedigree chart. So, listen to me carefully. Over here, if in every single generation you see affected individuals, for example, here affected individuals there, here also it's there, here also it's there. This parent, this child, this parent, again this child is there. That means it has to be a dominant disorder. It will be what? Dominant disorder. Okay, you just need one allele. For example, the condition is like this, my dear children, and if it is a dominant disorder, it will still express itself. You get it? So, yeah, that is dominant disorder. Now, how do you understand whether it's autosomal or sex-linked? So, if at all the males are the one that is usually more affected, it will be sex-linked. If at all it's the females who are mostly, if at all it's neutral, my dear children, it will be autosome-linked. It would be mostly autosomal. If at all males are the one that is mostly affected, it will be sex-linked. If at all it is neutral, you will normally see it is autosomal. Okay, got it? I hope you got it. Now over here you see one parent is affected, here also, so it is a dominant, and since both the male and the female are equally affected, I will say that it is what? Autosomal. One example of autosomal dominant disease given in your NCERT is myotonic dystrophy. What is it? Myotonic dystrophy. Now over here on the other hand, you have, in this generation you don't see this disease, in this generation also you don't see this, but their kid have this. That means it is recessive disorder, and still it is autosomal. Why? Because the males and females are equally mostly affected. So this can be an example of sickle cell anemia, autosomal recessive disorder, that is sickle cell anemia or hemophilia, sorry, not hemophilia, thalassemia, things like that. Okay, please remember myotonic dystrophy, very, very important. Okay, now let's the chromosomal disorders. First of all, there are 2 different ways in which you can get chromosomal disorder. One is aneuploidy and the second one is polyploidy. Aneuploidy means failure of segregation of chromatids during cell division cycle results in the gain or loss of a chromosome. So what happens usually when cell division happens? This set of chromosome goes there, this set of chromosome goes there, and the cell is divided. But over here, suppose there is non-disjunction happening, there is no proper division happening over here, my dear children. Do you think this segregation can take place properly? The answer is no. Which is why either you can get an extra chromosome or you may lose some chromosomes out there. So this is aneuploidy, which is normally taking place in human body. Now polyploidy is there, where failure of cytokinesis after telophase stage of cell division results in increase in the whole set of chromosome. That means what happened? Chromosome divided, but the cell division does not take place. Chromosome division, chromosomes got separated, the cells did not divide. So you have a whole extra set of chromosome out there. Okay, because why? Because the cytokinesis did not take place. This is usually seen in what? Plants, my dear children. So polyploidy is seen in plants. So you will be more asked about what? Aneuploidy. And under that you have different condition. As I said, trisomy. Trisomy is where, for example, I have 2 chromosomes. I should be having 2. I am getting 1 extra. So this is trisomy. Why I am getting 1 extra thing. Suppose I should have got 2, but I am getting only 1. The other one is not there. So this is what? Monosomy. Okay, so when an additional copy of chromosome may be included in an individual, this is trisomy. When you may lack one of the pair, it is known as monosomy. Now, you have 3 diseases: Down syndrome, Turner syndrome and Klinefelter syndrome. I think this is mismatched. It should come here and it should go there. Okay. So in Down syndrome, it is a case of trisomy 21. What do you mean by that? So 21st chromosome, my dear, autosome. Okay. 21st chromosome, you should be getting like this: 1 from one parent, 1 from another parent. But you're getting 2 from one parent, my dear children. So 1, 2, 3 chromosomes are there. And which is this chromosome? 21st chromosome. So you have trisomy of 21st chromosome, which causes Down syndrome. Extra chromosome is there. So how many autosomes do we have in general? 44 of them are autosomes. And then, as I said, is this XX or XY condition, right? Generally it is like that. But now in this case, you have how many chromosomes? 45 autosomes are there. 1 extra autosome is there. Plus it can be XX, that means it's a female who is having Down syndrome, or 45A plus XY, that means it is a male who is having Down syndrome. What are the symptoms of all these diseases? Very, very important. They have short stature, round head, furrowed tongue as in the tongue looks like this, broad palm crease, okay, mental retardation and delayed development. Short stature, round head, furrowed tongue, okay, broad palm crease and mental retardation and also delayed development. This is the condition, my dear children. Okay, so don't write this is wrong. Okay. So what would be it? It will be, my dear children, 45A plus XX or 45A plus XY. Cool? That's all. Now, this is known as karyotyping. This is known as karyotype. Okay, how many chromosomes or what is the thing? Now, next is Klinefelter syndrome. So, in Klinefelter's, you should be having X and Y. X coming from one parent, Y coming from another parent. But you have one more X out here. So, this is again trisomy condition. And how do you write the karyotype over here? So, autosomes remains the same, 44 of them. And then you have XXY condition. 44 autosomes, 45, 46, 47 chromosomes in total. Here also you have 47. 45 them and 2 of them, 47. So, here also you have, but it is in the sex chromosomes. Okay. Since it should have been a male, but now you have an extra X chromosome, my dear children. They will have masculine appearance, but with gynecomastia. What do you mean by gynecomastia? That is, breast development will be there. And this person will be sterile. They would not be having, what do you call, the spermatogenesis happening, because they have incomplete secondary sexual development. Okay, they would be having incomplete secondary sexual development, which is why they would be sterile. They will be having masculine appearance, but with gynecomastia, that is breast development. So either you write like this, or you can also write like this, 47. Don't specify autosomes. Just write 47 XXY, or you write 44A XXY. Either way, if you are writing like this, you have to mention A also. Okay. Now, Turner syndrome, the last one. This is where you needed to have XX condition, but there is no chromosome coming in over here. So, it's XO condition. Monosomy is happening over here. Again, how many autosomes are there? 44 autosomes are there, but it is XO over here. Or you can write in total how many you have? 45. You have 45 and then you write XO. You can write like that also. 45 and then XO. Otherwise, you mention the autosomes, 44 autosomes and then XO condition. It's caused by missing one X chromosomes in the female. There's one miss of X chromosome in the female. Features over here, since again, if both X chromosomes were there, obviously she should have been fertile. But over here, she would be sterile. Due to what? Rudimentary ovaries. The ovaries are not well developed. As I said over here, the males are infertile. Why? Because secondary sexual characteristics are not developed. Similarly, here rudimentary ovaries are there. The ovaries are not well developed, which is why they will be sterile. Lack of secondary sexual characteristics and short stature would be there, and they would be having some masculine feature as well. in Turner's syndrome. So this is Turner syndrome, this is Klinefelter syndrome. I hope you got it. And with this, my dear children, we end it. Here is a list of diseases: color blindness, hemophilia, sickle cell anemia, phenylketonuria, thalassemia, cystic fibrosis, myotonic dystrophy. All these disorders are Mendelian and rest of them are chromosomal disorders. So if at all you are asked to write a note on chromosomal disorders, you should be able to write it. And with that, we end the whole session. I hope in some ways it had helped you really well. If it had, definitely put it down in the comment section. If you have any doubts, put that also in the comment section. I am here to help you out. Wishing you all the best. Lots and lots of love. Bye bye. Take care.